Good morning, students. Today we will start with or discuss with the structure Megasporangium. The Megasporangium is also called ovule. Right? This, let's see the structure of ovule. In majority of angiosperms, the type of ovule is anatropous. So there are half a dozen of types of ovules. Among them, this anatropous type of ovule is very common. So hence, we will see the structure of only anatropous ovule. Later on, I will discuss about the different structures, okay, or different types of ovules. Right. So we'll see the anatropous ovule. So in anatropous ovule is covered by two integuments. The presence of two integuments is a condition we call bitechnic. So this is a inner integument and this is outer integument. So these two integuments later they form the seed coat of the seed. Okay. Integuments form seed coat. Right. So the ovule is connected to the placenta by means of funicle. So this is a funicle, and running through this funicle, okay, there is a structure inside, so which results due to the fusion of the ovule with the placenta, and this is called wrap. Now inside the ovule, this all empty space is not actually empty space, it is filled with the tissue, and the tissue is called nucellus. Embedded inside this nucellus, we find a structure called embryo sac. So this embryo sac is homologous to the female gametophyte. The female gametophyte is much reduced in case of diploid plants, even in case of monopod plants. In case of angiosperms, the embryo sac is reduced, okay, and it is present inside the ovule, surrounded by a tissue called nucellus. Right. Now, the one end of this ovule is called chalaza. The opposite end of the chalaza, where there is an opening, is called micropyle. Right. Inside this embryo sac, there are three cells present towards the chalaza, and these three cells are called antipodal cells. And inside the embryo sac, separately you will find, okay, two nuclei. So these are called the polar nuclei. And present towards the micropyle, we have total three cells here, okay, and these are called synergic cells. So this is, okay, a synergic cell, and between these two synergic cells we find another cell, what I call it egg cell. is called XL. Okay, so now these that is one XL plus two synergic cells together, they constitute something called the egg apparatus. Okay, so inside the synergic cells we also find finger-like projections, okay, whom we call them the filiform apparatus. They are called filiform apparatus. So total we find two synergic cells, one egg cell between them, two polar nuclei and three antipodals. Always whenever we have to draw the diagram, the three antipodals have to be drawn towards the chalazal end, and the synergic cells and egg will be always present towards the micropylar end. Right. So this is the general structure of this anatropous ovule. Right. Now, but how this anatropous ovule developed? Okay. So we know that this ovule is present inside the ovary. Okay. So when we see the structure of a pistil, we may find okay, so this is ovary, and whatever that space present inside is what we call the locule, and there is a placenta here. Attached to the placenta we find these ovules. Okay, so but how did these ovules have been developed inside the ovary? Already in the last chapter we have studied, after fertilization the ovary will develop into fruit and these ovules will develop into seed. Now let's see how this Megasporangium has been developed, and that process of developing Megasporangium is called Megasporogenesis. Now let us see the Megasporogenesis. Right. So this, the definition as it goes, the formation of megaspores from megaspore mother cells, okay, is called megasporogenesis. Is called megasporogenesis. Okay, so right here the megaspore mother cells, I call them MMC, megaspore mother cells, in short abbreviated form I call it MMC, and these megaspore mother cells will be actually diploid in condition. So they are diploid, and these megaspores will undergo the process of meiosis, okay, 1, and this meiosis 1 results in the formation of, okay, two, right, megaspores. So I can call it a dyad, okay, so that's called a megaspore dyad, okay, or else you can take like a megaspore dyad, okay, right. So there will be two in number. Now this, each one undergoes one more meiosis, and that results in the formation of, okay, megaspore tetrad. Okay, that consists of totally 4 cells. Okay, out of this, now the megaspore, the ploidy of this megaspore, whatever that the 2 megaspores, they will be haploid in condition because they have undergone meiosis 1. We know the meiosis 1 itself is a reductional division. Itself is a reductional division, and that undergoes, okay, the reductional division, the megaspore mother cells, and results in the formation of megaspore dyad, which are already haploid, and they again undergo the meiosis II and results in the formation of Megaspore tetrad. These are also haploid in condition. Okay, so here out of the four, what I call megaspores, three megaspores, okay, degenerate, right? Only one megaspore, okay, develops into the Megasporangium. So this one develops into the Megasporangium. So out of the four, three megaspores will degenerate, only one megaspore will develop into Megasporangium here. Okay, so such development, okay, so such development of Megasporangium from, okay, one megaspore is called monosporic development. Is called monosporic development. So let us see how the process of this monosporic development, okay, takes place. So here we'll see the first one, the megaspore mother cell. So this megaspore mother cell is surrounded by nucellus cells. I already told you that the female gametophyte is embedded in the nucellus tissue. So here this is all the nucellus tissue. So this is nucellus, okay, and at the center you find one very clearly, okay, conspicuous cell, and this is called the MMC, the megaspore mother cell. So this megaspore mother cell will undergo the meiosis I, and that results in the division of the megaspore cell into, the megaspore mother cell into two daughter cells, and those two will be haploid in condition, and they are called megaspore dyad. You can see here. So this is the megaspore dyad. The megaspore dyad, and already these two cells are the haploid in condition. Now this megaspore dyad will undergo the next division, that is meiosis II, and each cell undergoes, okay, meiosis II and results in the formation of two more daughter cells, that total together will be 4 megaspores. So 1, 2, 3 and 4 megaspores are formed. Of course, all this takes place, okay, at the center of this nucellus. So because of that, they are all surrounded by nucellus tissue. Okay, so this is still nucellus. So the nucellus tissue here, and you can see all these are megaspores. So it's called megaspore tetrad, and all those megaspore tetrads are haploid in condition. Okay, now I told out of these, what I call the megaspores, out of the 4 megaspores, okay, 3 will degenerate and only 1 will develop. So here that is what I am mentioning here. So here 3, okay, megaspores degenerate and only 1 remains. So this is 1 megaspore, that is haploid. So it consists of only 1 nucleus and it is haploid. And this one will undergo, okay, mitosis. Of course, the mitosis is not followed by cytokinesis, it is followed by only karyokinesis. So I call it okay, free nuclear division. free nuclear division takes place, which I mean only the nucleus will divide, whereas the whole cell doesn't divide. So that results in the formation of, okay, two nuclei. Of course, the type of division here is mitosis. So, okay, after first mitosis, okay, so this is what I mentioned, the first mitosis here. Okay, so this is the first mitosis, which results in the formation of two nuclei. I told you already it is a free nuclear division, which means only the nucleus divides, whereas the cytoplasm doesn't divide. And now this again, the nuclei again undergoes the second mitosis. It undergoes the second mitosis, and it results again. This is also a free nuclear division, and that results in the formation of, okay, four nuclei. So this is a two nucleate stage, and this is four nucleate stage, and now again it undergoes one more, okay, free nuclear division, and this is the third mitosis, okay, and that results finally, so as one nucleus divides, okay, one time, resulting in formation of two nuclei, as there are 4 into 2, you will get 8 nuclei. So this becomes something called 8 nucleate stage. Okay, so now this 8 nucleate stage will develop into the Megasporengium here. Okay, so let's see that how the 8 nucleate stage finally becomes a mature, okay, Megasporangium. Now out of these 8 nuclei, 3 nuclei will migrate to, okay, chalazal end, and results in the formation of, okay, the cell wall, and that takes place towards the chalazal. End. Okay, and the opposite end we have to take it as the micropyle. The opposite end will be having again 2 nuclei out of this 8 will migrate towards the micropyle and results in the formation of cell wall, and so, and inside also there is a development of finger-like projections and all, okay, and that results in the formation of synergic cells. So these 3 cells, okay, 3 antipodal cells, and these are the synergic cells. So this is a synergic cell, okay, 2 will be 2 synergic cells we have, and here you find one more, and this is, we call it the egg cell. Right, whereas out of, what I call 8 nuclei, so 2 became synergic cells, 1 became egg cell, and 3 became, okay, antipodal cells, and the remaining are 2 nuclei. They remain separately without formation of any, okay, cell walls, and these 2 are called, okay, the polar nuclei. And these 2 polar nuclei are the nuclei of the main cell, and this is called the central cell. So this is the complete, okay, formation of the Megasporangium here. So this is the embryo sac. This is the formation of embryo sac that is present inside the ovule here. Now let's see the ploidy of all the cells here. Now the antipodals, these all antipodals, 3 antipodals are haploid in condition. Okay, the synergic cells are also haploid in condition. the egg cell is also haploid in condition. Each polar nucleus is also haploid in condition. So the entire embryo sac is actually haploid. So in this you will find totally, okay, right, 1, 2, 3, 4, 5, 6, and this large central cell, the seventh one, altogether it forms, okay, 7 cell, okay, and there are 8 nuclei, okay, 1, 2, 3, 4, 5, 6, 7, 8, so it becomes 8 nucleate stage. So this is the characteristic feature of angiosperms. Okay, so that's I mentioned here, okay, as a note. 7 cell, 8 nucleate, okay, condition is a characteristic feature of angiosperms. So nowhere in the plant kingdom you find, okay, this condition with okay, 7 cells and 8 nucleate. So this, the largest cell is the central cell. Embedded inside the central cell you find 3 antipodals with their cell walls. All of them are haploid in condition, and the synergid cells are also haploid in condition, including the egg cell here. Okay, so inside the synergid cells we find one finger-like projection, like special structures, and I call them the filiform apparatus. So this filiform apparatus actually helps in guiding, okay, helps in guiding pollen tube. So helps in guiding pollen tube. So already I have mentioned that this one egg cell, okay, and 2 synergid cells always that present towards the micropyle, and this one egg cell and 2 synergid cells, okay, together they constitute, okay, egg right apparatus. Right. So this is how the process of, okay, megasporogenesis takes place.